better to do this one, then we'll do adrenal, all of adrenal next time, which is tomorrow, right?
Yes.
Okay, anyway, well, my name is Eldridge. I probably will sit down while I do this so I can manage the slides and because I'm old and I'm lazy. So, and I do hormone things, and that's sort of what we're going to be talking about. A lot of different areas, particularly in that are involved in reproduction and so on. So what I'm going to begin this with, and I don't really know all of the, I'm not familiar with all of the other coursework that you may have had prior to this time, but a lot of times when people talk about hormones, they're talking about those that interact with cell membranes and use second messenger systems, which is by far the predominant way that hormones and other cell signaling works. So I'm going to say a few things about steroid hormones, and which are quite unique, starting with the fact that steroids are able to enter into the cells because they are lipophilic, and in fact are not at all water soluble. So these hormones interact with the nucleus of cells, that is to say the genome. So I'm going to review some of that. You may have had this somewhere else, but I'm going to take the liberty of doing it again. And I'm especially going to contrast the biological, which means cellular effect differences between the different classes of hormones, of steroid hormones. And then I'm going to also say a few things with respect to pharmacology. Most of this is physiology, but I'll say a few things about pharmacology as to how hormones like these are chemically altered in order to allow them to be used as medication. All my talks have a little bit of what I call principles. These are the things, the main messages that I want you to leave here with. So most of what I'll be saying leads to these messages. Hormones work with specific receptors. That is to say, there is a protein chemical inside each target cell. In fact, that defines a target cell. A target cell is a cell that has a receptor for a hormone. This is very much like locks and keys. So hormones have specific protein receptors that trap the hormone, and that begins the action, the biology of the action. Obviously, there's a different receptor for each hormone type. But hormone action always starts with this kind of receptor interaction. Moreover, hormone drugs, see, hormones are natural chemicals in the body, but hormone drugs use the same receptors. Of course, receptor binding and response are two different things, and in fact there are a lot of antagonists of hormones that are used as drugs which block the action. That means they bind the receptor but don't have a response. So that blocks the hormone from doing something. But it's important to remember that side effects use the receptors too. In other words, a hormone may bind, a natural hormone may bind a whole bunch of places around the body. Well, a hormone drug is going to do that too. So for the most part, side effects are the result of the hormone doing the hormone drug doing the same thing that the hormones do. Okay. Now you can see here these are the typical examples of the classes of steroid hormones. And testosterone is a male hormone, estradiol is a female hormone, and you can see these molecules are very, very, very similar in structure. Obviously, the body knows the difference. And that's because the receptors are specific, but the receptors themselves are very similar. And you can see not just steroids like cortisol, progesterone, vitamin D, which is actually a steroid. These are all hydrocarbon bonds here, carbon-carbon bonds. So you can see testosterone has one hydroxyl group and a ketone group. This is not going to be soluble in water. Cortisol, progesterone, and also thyroxine, which is water soluble, happens to use the same receptor class as the steroids do. That is to say, nuclear receptors. So here's what these nuclear receptors do. The ligand, that would be the hormone, enters the cell. This is the cell membrane, okay? They enter the cell and they enter the nucleus, and they find a receptor molecule right here. And these receptors, when they bind the ligand, the hormone, they double up. They couple to another bound receptor. That's called dimerization. And the pair goes to a spot on DNA to bind literally to DNA and locate a spot for transcription to begin. So this whole process is leading to new mRNA, a new protein, a new product for the cell. That's how they work. So here's showed the dimerization binding to DNA, which is not quite, and I got another slide that shows you this, it's not quite at the start site. It's upstream from the start site, but RNA polymerase II is then activated to begin transcribing new message, which leads to a protein product that leads to expression of some biological action. That means the effect is not instant. It takes a little while for a cell to make new mRNA, new protein, and a new response. Now there's over 150 of these receptor type in this family. This is called a superfamily of nuclear receptors. Lots of them have been identified now by cloning, and the fact is that most of them don't bind anything that we know of, so they're called orphan receptors. Most of the steroid hormones have one or two receptors that they bind to. That is to say, it's a very specific kind of cell biology. There are other compounds, as I said, that bind to this family of receptors, including peroxisome proliferators and some bile acids and other kinds of intracellular compounds. And the key here is that the receptor itself doesn't do anything until the ligand, the hormone, binds to it. So what happens when the hormone binds to it is that the receptor changes its shape. Literally, it's a three-dimensional thing. Now, all of these receptors are single-chain proteins. And ironically, I think this is so cool, the androgen receptor is actually located on the X chromosome, which means females have 2 copies and males have 1 copy of the androgen receptor. So the X chromosome has a particular gene, which, as you can see, has lots of introns and exons, and then this becomes a message which is edited out, and you get a single large, very large, upwards of 1,000 amino acids for this receptor molecule, which means 3,000 bases, which by the time you include all the other stuff, you're talking 8,000 or 10,000 bases in that gene. Now the receptor molecules, this long chain, of course they fold up. They're not all just one long stretch. They fold up, and in fact, they form a little hydrophobic cleft. I said the steroid hormones are not water soluble. The steroids themselves are very hydrophobic, and therefore they look for a binding site and will stick very tightly to a binding site, which is itself hydrophobic. So this is the key that slides into the lock. But the lock itself is very hydrophobic, so the steroid sticks with very high affinity. This is nanomolar affinities. The receptor molecule has an area which binds the hormone, and that's called the ligand binding domain. Then there's a little piece here that binds DNA, and it's looking for a specific sequence of DNA. And then there's another domain out here that binds other proteins, and those are the activating factors.
Yeah, question. On a previous slide, since it's like since females have 2 copies.
Okay. You're gonna have to speak up because I'm old and don't hear well.
On a previous slide, since females have 2 copies,
Yeah.
do they change genetically, or do they kind of stay the same within everyone?
Oh, the question is, if females have 2 copies, is it the same copy?
Yeah.
Not necessarily.
So they do change within people.
Remember in cells, in female cells, only 1 X chromosome is active in that particular cell.
Right.
Remember? 1 is inactive, so that provides a greater variety of response. I'll say a few more things later. Androgens are active in females. Okay, well, estrogens are active in males too, but androgens are quite active in females. So androgen receptors are significant in females, yes. The impact that it has, of course, is that every ovum is haploid, just like every sperm is haploid. So the X chromosome that's passed to the embryo could be different from the one that's passed to the next embryo. But in males, they only have 1 X, so that X is going to get passed every time. That X is passed if it's a female. If it's a female, right? If the embryo's a female, that X is passed. Yeah. Here's just a little schematic that shows the example of this superfamily. In other words, the estrogen, and as I said, there are 2 estrogen receptors called alpha and beta, and where the hormone binds is very, very, very similar to all of them, and where DNA binds is very, very similar, not identical, obviously. And then this A-B region can be very different because that region attracts these other transcription activating factors that the cells use to promote transcription. So this is progesterone. There's an A and B of progesterone. androgen, mineralocorticoid, glucocorticoid. And this is the number of amino acids in those proteins. You can see here upwards of almost 1,000. Okay. Now, the receptors, as I said, they dimerize, which means that two receptor molecules bind only after the hormone has bound. In other words, they don't dimerize before. Each one binds one molecule of the hormone. Now, sometimes these receptors will bind to a different receptor. That would be called a heterodimerization. Sometimes they bind to their own. That would be a homodimerization. So sometimes, and it's not two hormones that are going to bind estrogen and androgen together, that sort of thing. Sometimes they bind with something else, as I said before, like retinoic acid or a peroxisome proliferator type of ligand. Okay. But this dimerization is necessary, and I'm going to show you why. Because each one is going to bind to the DNA. Each one is going to bind to a sequence on DNA. And so how does this work? When a hormone binds to the receptor changes shape. I told you that. When the receptor changes shape, a couple of fingers are kicked out from that receptor molecule. Those are called zinc fingers because there's an atom of zinc in the middle of it that holds it together. Each of these little letters is a code symbol for an amino acid. Okay, so this is a little, like what, about 10 amino acids, 12, 14 amino acids in this little finger that sticks out. These little fingers are not sticking out when the hormone has not bound yet. So they stick out like this. Okay, and the dimerization, and this is very cool. I want to borrow your right hand, please. Okay? Now, we have, see, our hands are the same orientation, right? Okay. So, close your fist, and I'll close my fist. So if these bind back to back like this, now stick these fingers up. Okay. And I stick these fingers up. Now, what happens is that this receptor molecule would be looking for a sequence. Wait a minute, they go this way. This receptor molecule is going to look for a sequence, and my little finger is this direction. Okay? Hers is going to look for a sequence going this direction. That's opposite, right? Okay.
Is this in the...
Why is that important? Because each receptor is going to bind to DNA and is looking for the same sequence, but it binds to both strands. In other words, one receptor binds, you notice right here, this receptor with the two zinc fingers is binding to this strand of DNA, while this receptor, which is stuck to it, turned in the opposite direction, is binding to the other strand of DNA. But it's looking for the same sequence, of course, because it's the same receptor. But you know the DNA coils around and goes opposite directions, right? The code goes in opposite directions, and they're complementary to each other. So the receptor binds to both strands of DNA, the same sequence, but each going in different directions, but they obviously have to be located very close to each other as the two strands are passing. Right? That's that. Okay, let me see. And that's called a palindrome. So here's what's cool about it. Estrogen receptor is looking for this specific sequence. This is bases. Adenine, guanine, guanine, tyrosine, okay? And the other receptor is looking for the same sequence, but it's going the other direction. But of course, since this is DNA, the complement bases are also going in the opposite direction. Okay? So the receptor, it takes two receptors to find the same sequence close to each other. The little N's mean nonsense. It means it doesn't matter what that code is. So you've got a sequence here of 12, 15 bases that the estradiol homodimer needs to bind to. And each hormone, each receptor needs to bind. And here's what's interesting about this further. The glucocorticoid receptor is looking for the same sequence except two are changed. One of the old principles of biology is that biology likes conservation. That is to say, it manages to function with minimal change. So what you've got here is a completely different receptor binding to DNA, and it's able to function by looking for the same sequence with only two of the 6 bases changed. And that works. Glucocorticoids are not estrogens. And thyroid is looking for an 8-base sequence, which is the same as estrogen plus 2 more. So I'm not telling you this because I want you to memorize all this stuff. I'm showing you this as to how life manages to function with very minimal effort, but yet it's a very specific kind of activity. All these hormones are different, they do different things, and obviously the receptors locate the right spot and not the wrong spot and make different things happen. Because a cell might have an estrogen receptor and a glucocorticoid receptor and a thyroid receptor, and all 3 of those hormones have some biological effect on that particular cell. But they're all binding to a different, slightly different location in DNA. Obviously that means you're going to get different message and you're going to get different protein products. Now, as I said, hormones promote transcription. These hormones promote transcription. But the receptor doesn't bind at the start site. These receptors bind on DNA some distance upstream from the start site. So what happens? What's the connection? The connection is that the receptor, this would be the dimerized receptor. The connection is that, and not everybody agrees on this, but this is what I think happens. This black curve here would be DNA. And so the thinking is that the receptor complex binds upstream and begins to attract these activating factors and other co-regulators. These are proteins in the cell that will come to attach to the receptor when it's bound to DNA. And when that happens, the more of this that can be accumulated, the more transcription you're going to get of new message. But that the DNA sort of curves around and attracts a large complex of activating factors. And this is what makes the hormone, which activates the receptor, which then binds the DNA, which attracts all this, what makes it all promote new message. Okay. So let me mention now quickly what a few of these receptor complexes do as far as the biology of the tissue. Vitamin D, I don't know if you've heard this already. You've had vitamin D? Not yet. Okay, you will. Dr. Howlett. Vitamin D is important for calcium mobilization in the body. And this is just another cartoonish thing out of a textbook showing that the vitamin D receptor, which binds not to another vitamin D receptor, but a retinoic acid receptor, okay, binds to DNA. This would be the vitamin D response element. Don't pay attention to there's all sorts of code names and acronyms, which then eventually promotes transcription. But the important thing there about vitamin D is that it doesn't dimerize. This is a heterodimer. It binds to the retinoic acid receptor, not another vitamin D receptor. No matter. It's the way they work. What vitamin D does, as I said, is to promote calcium mobilization, particularly in the GI tract, to absorb calcium, dietary calcium. We don't, almost everything in the body needs a transporter. I mean, we just talk generally about how things move around. Glucose needs a transporter. Obviously, this is why insulin is important. Calcium needs a transporter. Sodium needs a transporter. Everything needs some kind of transporter protein that will carry it across the cell membrane. And so dietary calcium is not absorbed. And vitamin D is necessary to stimulate the transporter protein that will carry calcium from the lumen of the GI tract into the blood. But vitamin D happens to be also active in all of the other, in fact, the vitamin D is in every tissue in the body because all cells need calcium. Now, estrogen is a far different kind of hormone. The responses to estrogen are far different. Estrogen tends to be what's called a proliferative hormone. That is to say, it promotes mitosis in the target cells. And that's important to know because cancer itself is a mitotic problem. In other words, a cell type that's dividing rapidly, more rapidly than the body can handle. And so this means that estrogen responsive tissues that are dividing, their tumors of those tissues are also going to be promoted by estrogen. This is also true of androgens. So that's why breast cancer, uterine cancer, prostate cancer are all promoted by the respective steroids, and why therapy that antagonizes that hormone class, is going to have some therapeutic value. See, now I talked just a minute ago about vitamin D. Vitamin D is not a proliferative hormone. The action of vitamin D is not proliferative. See? So there's a difference. Of course, they use their own mechanisms, but there's a difference of the kind of proteins and the kind of cellular response. Progesterone, which is also a steroid, well, in women, it exists in men, but there isn't any known action in men. Progesterone moderates and to some degree antagonizes the estrogen action in women. But in addition, many steroids do other kinds of things. So it's said the vitamin D, but even estrogens and androgens do other kinds of things than stimulate mitosis or tissue growth. And so even nerve and brain and muscle and so on are target cells for these hormones, but those cells aren't dividing. Okay. Estrogen, as I said, has two receptor types, and this has become a very, very important field of research for pharmaceuticals, because the two receptors are not, many of the cells have both receptors, but the populations of the receptors and the effects of the receptors are different. And I don't want to draw too sharp a distinction, but estrogen receptor alpha tends, and that's the original receptor that was discovered, alpha tends to be found more in the tissues that are concerned with reproduction, while the beta receptor is found in lots of other places around the body. Now, as I said, don't assume that that's a sharp demarcation. Many tissues have both receptors, but the two receptors, obviously, both receptors bind estradiol. Both receptors bind natural estrogens. Sure. But still, there's a slight difference in what they bind. And the pharmaceutical industry has come up with some chemicals which bind more tightly to one than to the other. And furthermore, the kinds of message and protein products that come out are different from one versus the other. So there is a lot of active work now to try to design drugs that can be an estrogen-like agonist at a spot where agonist is beneficial, and an estrogen antagonist at a spot where you want to antagonize, where you don't want estrogen action. Okay? I'll say a little more about that later. So, as it shows here, even the DNA binding domain is not exactly the same. It's 96% one compared to the other, and the hormone binding domain is only 60% identical. So this leaves plenty of room for some kinds of chemicals that would maybe bind to one and not the other, or one more actively, tightly than the other. A lot of work here. Estrogen, as I said, is a proliferative hormone class. So the effect here is to proliferate. The endometrium is the lining of the uterus. So it causes a proliferation, that is to say mitosis, a growth of the uterine lining. It also hypersensitizes the myometrium, that's the muscle part of the uterus. The uterus is mostly a smooth muscle organ, and that smooth muscle can contract, and estrogen promotes that. And I'll contrast that to progesterone in a minute. It also, estrogen stimulates a cervical mucus from the cervix that is thin and watery. And I'll contrast that to progesterone. Estrogen has a negative effect on pituitary hormone secretion, FSH and LH. It's a negative feedback to suppress FSH and LH, which is the principle of oral contraceptive. Estrogen also in mammary tissue promotes the ductal epithelium. Now, not the milk-producing part of the mammary gland, but there's a ductal system that transports the milk from the factory, the lactiferous gland, to the surface. And this ductal system is promoted to develop and grow by estrogen. This is important for two reasons. One is that most all malignant cancer of breast is ductal epithelium cancer. In other words, this is the cell type that turns malignant. Therefore, estrogen would be expected to promote that. Therefore, an estrogen antagonist would be expected to be therapeutically useful. Okay. And of course, any of those cells that spread around the body, the same thing would hold. Okay. The other point I want to make about this is that mammary tissue is the same in males and females at birth. In other words, the reproductive systems are different in males and females, but males have the same mammary tissue, therefore estrogen effect would be the same in males. This is a clinical point. Estrogen effect would have the same response in males as it would in females. Males have all of the same equipment to respond to estrogen. Estrogen has a very unique effect on the skeleton. As children grow, reason children grow taller is that the long bones have a piece of cartilage that continues to divide as the children throughout the childhood life. So long bones in the legs and in the arms and so on, this cartilage keeps dividing and then it fills in with bone, but the cartilage stays there and keeps dividing and keeps dividing. Well, at some point that closes off, that stops, and the cartilage that remains becomes bone. That process is promoted, that closure is promoted by estrogen, not androgen. This is why, and I got another chart to show you, this is why boys, on average, wind up taller than girls, on average. Estrogens are anabolic. Anabolic means preservation of protein. The opposite of anabolic is catabolic, where you're tearing down protein to make energy. I'll talk about that a little bit too. Estrogen increases HDL cholesterol synthesis in the liver, average women have much higher HDL, which is the safer form of cholesterol. Women on average have much higher HDL than men do. Okay, a couple other effects. So here's the growth thing that I wanted to point out. If you take the pediatric growth charts and you plot just the average, you know, those growth charts always have a range on them and so on, but if you plot just the average numbers of children in the United States as they grow, you find that boys and girls are on average the same height up till the age of 10 or so. And then on average, girls get a little bit of a growth spurt. That's because of puberty starting. But shortly after that, the growth curve flattens out. While in boys, the growth spurt continues and continues and continues until it flattens out at a later age. In other words, basically speaking, the average girl's height has topped out at, say, first year of high school or so, second year, while most boys will continue to grow throughout high school or later. This is the estrogen effect. Would estrogen do the same thing in young girls with premature puberty, or estrogen exposure in young do the same? Yeah, it sure would. And furthermore, very interesting thing, there is the third little point I have here. The reason we know that this is all true is that there is a genetic syndrome in a very few small population of males where they lack the enzyme that converts androgen to estrogen, so they have no estrogen of their own. All males normally have some estrogen. They have a lot of androgen, which overrides the estrogen pretty much. But they have a little bit of estrogen. There's a syndrome in which these males are unable to make any estrogen at all because they lack a specific enzyme that converts estrogen, CYP19, this aromatase enzyme. The interesting thing is, these males have epiphyseal cartilage that never closes. It never closes. That doesn't mean they get to be 8 feet tall. Your growth will stop anyway, pretty much, by age 18 or 20, even without any estrogen. But the epiphyseal cartilage is still there in these adult males. So we know for a fact that estrogen closes that. What it does is it stops the mitosis of the cartilage, and that cartilage fills in with bone eventually. Now, as I said, estrogen does a number of other things in the brain and other parts of the body. In other words, it does more than proliferative. So it's known, this is obviously through cloning technology and so on, it's known to promote a whole bunch of other CNS actions, for example, that affect behavior. So do androgens. I can find the same, that would be different responses. Okay. Females also have progesterone. Males have progesterone too, but there's no known biology that in males that relies on progesterone. All of the progesterone actions occur in females in tissues that are female active, shall we say. So, progesterone, for example, stimulates the lactiferous gland part of mammary tissue. This is the milk-producing part, not estrogen. Progesterone does this. And it requires a very high level of progesterone that's found only in pregnancy. So that, in other words, histologically, the mammary gland of a woman who's had a child appears differently from a woman that has not had a child. Okay, so normal milk secretion, that factory, is developed in pregnancy, but it's not developed before pregnancy. And that has to do with levels, hormone levels. Progesterone levels get very high during pregnancy. That comes from the placenta. Okay. Progesterone potentiates the action of estrogen on LH and FSH secretion. And so most of the contraceptives contain progesterone as well as estrogen. There are other reasons for that too. I'll get to that. Now, those things that I talked about estrogen doing in the uterus are changed and to a certain extent opposed by progesterone. The menstrual cycle of women, the first half before ovulation is mostly estrogen dominated. After ovulation, there's a lot of progesterone added to that, the second half of the cycle. So the first thing that progesterone does is that endometrium that is stimulated by estrogen is transformed, it's changed to what's called a secretory endometrium, lining of the uterus. And this is critical because the embryo needs that secretory transformed lining to implant. Okay, so if ovulation occurs in the middle of the menstrual cycle, fertilization occurs a day or two after, that implantation has to take place within about the next 10 days, and the endometrium has to have been transformed by progesterone to be the type of tissue that can receive that embryo, the implantation. By that time, the zygote is a little blastocyst stage. It's many, many cells, hundreds of cells. While estrogen promotes uterine contractility, smooth muscle contraction, progesterone suppresses that. So during pregnancy, the high progesterone keeps the uterus relaxed and not contracting. You don't want to deliver the baby until the baby's ready to be delivered. So the high progesterone keeps the uterine muscle quiet. And then at the very end of pregnancy, labor is started when progesterone starts to drop and the estrogen is able to start to cause myometrial contraction. So in this case here, progesterone is doing the opposite of what estrogen does. And progesterone, I said that estrogen produces a thin, watery cervical mucus. progesterone promotes changes that cervical mucus to a thick cervical mucus, which appears to be somewhat of a barrier to sperm passage. So when I talk about drugs and reproduction, I'll talk about some contraceptives that use only progesterone. Well, how would they work? Well, one thing they probably do is to thicken the cervical mucus, which tends to retard sperm passage. Okay. And as I've said a couple times already, there's no specific role because males don't have functioning mammary tissue, males don't have a uterus. Those are the two main actions of progesterone in the body, and also only females are going to be using those actions. Okay. Androgens, and another point. I'll jump into pharmacology here and there. Estrogen drugs are going to do what estrogen does, right? I mean, progestin drugs will do what progesterone does. Androgen drugs will do what androgen hormones do. Okay, so androgens, of course, promote the male reproductive system, and androgens are much more anabolic than estrogens are. And this is a skeletal muscle kind of effect on metabolism and on the muscle tissue. So would this effect be expressed in females too? Oh yeah, sure. Anabolic steroid abuse is the same in females as in males. That is, it has the same usefulness in females as it does in males, because it'll do the same thing. I probably should change this picture because most of you guys are not even sure who Mark McGuire was, right? Or would recognize the picture. This is not a Photoshop picture. Way back, when he started admitting that he had been using androgens, and he admitted that he used natural androgens. He posed for pictures like this, Androgens have an effect on hair follicles. All hair follicles are affected by androgens. Most of the hair follicles, say from the nose on down, androgens stimulate the hair follicles to grow hair. Males and females. Females have some androgen. It's a matter of, the difference in males and females is a matter of how much or how potent the androgen. Males tend to have a lot of testosterone that females don't have. And so that promotes hair growth on the face and body and so on that women normally don't. But women can grow hair in all the same spots that men grow hair. So from a clinical standpoint, that could represent some kind of abnormal situation, that is an abnormal source of androgen in women. This would be a tip-off. Of course, androgens also cause loss of hair in some of us who are very fortunate, at least if you ask my wife. She likes my hairdo. So it also causes loss of hair in individuals, again, in males and in females. It's a question of how much androgen you got. Okay. Of course, the vocal cord effects. Androgens do support spermatogenesis a little bit. That's not a big deal. Androgens also stimulate red blood cell production. The one legitimate use of anabolic steroids is that they promote red blood cell production, and particularly in people who are suffering from some kind of degenerative disease. The average red cell count, the average hemoglobin level of the adult male is 10% higher than adult female. This is because of androgens promoting red blood. So the anabolic steroids, for the most part, are legit drugs, even though they're scheduled drugs. They're legit drugs that are legitimately used to promote red blood cell production. And even low levels will work in children and in women, very importantly. Now, the major male hormone from the testes is testosterone. This is the hormone in blood, made only by the Leydig cells in the testes to any large degree. Many of the target cells for androgens convert this testosterone to an even more potent, higher affinity, more potent androgen called dihydrotestosterone. And what happens is this double bond at the 4 or 5 position gets reduced. And so that's called 5-alpha dihydrotestosterone. As I said, this is even higher affinity androgen inside the target cells. There's not a lot of dihydrotestosterone in blood, but male target cells tend to convert. I don't say male target cells. Target cells in males tend to convert, and to some extent they would in females too if they had any testosterone. So androgens are important in males at 2 stages of life. One is the prenatal stage. The default development in fetuses, the default development, that is if there was no gonadal tissue at all, the default development would be female phenotype. But the presence of androgens from the fetal testis causes development of the female system, of the male system. Okay? So, let me go. So, testosterone, which would be the lower affinity androgen action, includes some of the internal structures, while the dihydrotestosterone, it's the same androgen receptor, different ligand though, causes development of the external structures and the prostate. So that's during fetal development. So at puberty, the adult development of males requires a lot of testosterone and also some dihydrotestosterone. And here's something that I want you to notice. Again, this leads to pharmacology. Notice that the skeletal muscle and skeleton type, or skeletal thickness and growth that males have, adult males have, is a testosterone type of response. That's why anabolic steroids, which are pretty weak, still work. While dihydrotestosterone is involved with prostate and with body hair. So that means that drugs that block dihydrotestosterone production, drugs that block the 5-alpha reductase, are effective for prostate problems. There's benign prostate problem, and then there's malignant prostate problem, and also for body hair growth in both genders, okay, and also for baldness treatment. Okay, so there's a difference here between 5-alpha dihydrotestosterone actions and actions that are sufficiently promoted by testosterone alone. And the anabolic action is one of the most important that testosterone alone can take care of. Okay. All right, so I'll wrap this up with a couple of other little things that really is pharmacology. Steroid hormones, as I said, are lipophilic. They're not soluble in water. So if you want to administer a steroid hormone orally, it's not going to work very well, because this kind of compound will be conjugated as esters, actually. They all have hydroxyl groups, so you can make an ester out of them, and that makes them water soluble, and then they're peed out. And so they don't last very long in the body. So there are two things that I'll tell you about. One is, because they're lipophilic, they can be injected. But they're better injected in an oil vehicle. Why? Because they're already soluble, better soluble in oil than they are in water. So the oil-based vehicle will retain that hormone in the vehicle for a long period of time. So the absorption rate is very slow. And if you create an ester linkage to a long chain fatty acid, then the absorption rate can become very, very, very slow. So there are steroids for injection that will last for months. This medical center, the OB/GYN department gained a big piece of favorable publicity when they discovered that some women who were at danger of miscarrying during late pregnancy, and I told you why they would be miscarrying, because progesterone, that ratio of estrogen and progesterone has to be maintained high progesterone. So what they were able to do was to figure out that these patients were running low on progesterone at 7 months, so on. So what they started to do was to inject progesterone into these patients, but they only had to do one injection. You could do a progesterone ester in oil, and that would be absorbed over a long period of time, very slowly. Because the partition, you see, if the hormone is soluble, more soluble in the lipid than it is in the water, then that partition favors the oil side. So it's going to be taken up very, very slowly from that depot. Okay. But once once it gets to the body, that ester linkage is broken very easily, so you're back to the natural hormone in blood. So it's a way to deliver natural hormone at a very slow rate. Very clever. So estradiol, for example, can be tagged to this complex here. This is valor cipionate, cipionic acid, as a fatty acid. And progesterone can be tagged with this side chain here. And testosterone can be tagged with this side chain. These are just fatty acid side chains. And of course, they're innocuous too. They'll just enter into the fatty acid metabolism in the body. No problem. So it's a very safe medication. It has to be injected IM, intramuscular, which is not comfortable. Another reason for not doing it very often. But it still is absorbed very slowly and very effectively, and it's very safe. And yes, there are anabolic steroids that are also made this way. So that's what you do to prolong absorption rate of a natural hormone using an oil vehicle for injection. You would not give this subcutaneous, you would not give this in IV or anything like that. Now, for oral activity, you got to do something different. What happens to everything that you eat that goes into the GI tract, that's absorbed in the blood? Where does that blood go? First of all,
Liver.
goes to the liver, yeah. Every drop. All of the blood from the GI tract is collected into veins that go to, that's called a first pass effect. The liver has first crack at everything that you take in through the GI tract. Okay. Well, the liver is programmed to conjugate these hormones as water-soluble esters to pee it out. Well, heck, that's not what you want. You don't want the liver to remove the stuff before it even gets into the rest of the body. So, the estrogens and the other androgens and so on are chemically modified to interfere with that conjugation system. And that's done by tagging these molecules with some other, I'm just telling you what, or I'm showing you in words what I just told you. By tagging these molecules with an extra change or an extra group somewhere that interferes with the estrogen, or that interferes with the hepatic metabolic enzyme system. But obviously, you don't want to interfere with the hormone binding part. You still want the hormone to be a hormone. So there's some interesting chemistry that was done there so that you could have a molecule which still fits the receptor like it needs to do, but it doesn't fit the enzyme in the liver that's going to conjugate it as a glucuronide or a sulfate or something like that. So that's basically just a steric hindrance with some kind of bulky side group. And so here's what's done. This stuff doesn't come off. When I talked about the injected steroids, those are esters, and the esterases will cleave that so you're back to the native hormone. But these little groups don't come off. These are carbon-carbon bonds. So estradiol, for example, has an ethanyl group at the 17 position. And that's sufficient to interfere with hepatic conjugation. Ethinyl estradiol is the principal estrogen in oral contraceptives. In other words, 1 pill a day is enough. If it was native estradiol, you'd have to take it every 2 hours, 4 hours. You know, not gonna work. So ethinyl estradiol retards that metabolism and clearance sufficiently that you can have 1 per administration per day. That's the ethinyl group, triple bond carbon. Progesterone has this 6-alpha methyl group here, the 6th position. So that's good enough to block progesterone clearance. And down here is an orally active androgen, 17-alpha methyl. So, and a floral group at 9. So this compound is an orally active androgen, not nearly used as much as orally active estrogen. Okay. But it still works. And the principle here is that by retarding the clearance and metabolism, you have a much higher dosage on board for a longer, in other words, it'll circulate for a long period of time. And the liver is not able to get rid of it as quickly. Here's some orally active steroids, adrenal steroid, prednisone, which is a metabolite of cortisone, not cortisol. The ketone at 11 has a double bond at 1,2. That makes this compound orally active, but only a little more orally active than cortisol. But this is a rather weak but very popular oral glucocorticoid. You can make a more potent oral glucocorticoid by putting on this 9-alpha fluoro and a 16-methyl group right here. Here's a mineralocorticoid with a 9-alpha fluoro. Okay. The principle in all of these is to retard clearance and metabolism. Okay. So tomorrow I'll be talking about a whole bunch more of the adrenal gland, of putting all that together, and then later will be stuff about reproduction. But this is kind of the basic steroid part. All right, question, I thought I saw something. Yes, sir.
Dr. Howlett, she's not giving us a quiz for this section, so I was wondering, she's giving us like some take-home stuff to do. So I was wondering for your section, are you giving us a quiz?
Well, I think I'm going to talk with Dr. Howlett. We're probably going to give you guys something. We're going to have to ask you to write a little bit.
Okay.
I'm not afraid of that.
No, I was just trying to wonder if you were giving, like, yeah, she's already told us that she.
Yeah, I'm not going to give you a bunch of multiple choice questions, no.
Okay.
I don't think so. Maybe a few.
That'd be all right.
Wait, so is it
In
take
class
home
or
it's
take
gonna
home?
be like a quiz in class?
Well, I think it'll all be take home, I think,
Okay.
yeah. I'd rather see you guys write. I dislike multiple choice questions. I mean, I understand various kinds of board exams and SATs and everything else, what else is, in fact, grading essays is subjective, there's no two ways about it. But with this kind of thing, I mean, what I'm looking for, A, do you understand something? You know, did I show you something, or did you read something, did you find out something that you understand now that you didn't understand before? And then secondly, can you translate that into a little bit of an application? In other words, now I understand this, therefore, something either clinical or experimental as a result from that. Yeah. Tomorrow, no.
Y'all just already-
Tomorrow, they have a faculty meeting in here, so I'm pleased to miss it, so I can.